Hello everyone. In today's video, we will study the remaining part of megasporogenesis. We have covered the major portion of it in our previous video. So in this video, we will study all the cells which are there in the embryo sac. Till now, you already know that embryo sac is also known as female gametophyte. And you already know that whenever embryo sac is formed, it has 7 cells. So these are the 7 cells which I am drawing. 2 cells in the center, 3 cells at the upper end. Again, important thing to note down is that micropyle end is located here and chalaza end is located at the top, because we are talking about an embryo sac that is present in an inverted ovule. So let's name the cells from micropylar end to the chalazal end. The 3 cells at the micropylar end, they are known as egg apparatus. Now in egg apparatus, you can see that 3 cells are there with their nucleus, and each nucleus is haploid in nature because megaspore was formed as a result of meiosis. So megaspore was already haploid in nature and it went into 3 consecutive mitotic divisions. That means the ploidy level will remain haploid only. This central cell will be known as polar nuclei, and the 3 cells here at the top, they will be known as antipodals. So you can see that 3 cells are there at the chalazae end, 2 cells are there at the center, and 3 cells are there at the micropylar end. Here the bigger cell is the egg, and this is actually very important because it is named as female gamete. The other 2 cells, this one and this one, they are known as synergids. So in the embryo sac, 3 cells are there at the chalazae end, 2 nuclei are there at the middle position. They are enclosed in one cell. So this altogether is a cell and it has 2 nuclei. So 3 nuclei at the chalazae end, 2 nuclei at the center, and 3 nuclei at the micropylar end. So overall, how many nucleus are present? 8 nucleus are present. And how many cells are there? 1, 2, 3, 4, 5, 6 and 7. How many cells are there? 7 cells are there. So in an inverted ovule, the embryo sac is generally 7-celled and 8 nucleated. You must learn the names of all the cells. Along with this, figure drawing is also important. One more term is important before we sum up with our megasporogenesis, and that is monosphoric embryo sac. Most of the times question comes, what is monosphoric embryo sac? So let me explain this. Monosphoric means a single spore. Now you have already seen that through megasporogenesis, 4 megaspores were formed. This was the micropylar end, this was the chalazae end. All these 3 megaspores, they degenerated and only the chalazae one was functional. So only this megaspore was functional, only one was functional. And this embryo sac, it actually developed from only 1 megaspore at the chalazae end, right? What happened actually? This was our 1 megaspore with the haploid nucleus. It went into 3 mitotic divisions as told by me in the previous video. So first mitosis will produce 2 nuclei, Then second mitosis will again produce 4 cells, because 2 nuclei will form 4 cells, and when third mitosis will happen, then from 4 how many nuclei will be there? 8 will be there. Now we have already seen how these 8 nuclei they have arranged in the embryo sac. So embryo sac developed only from one megaspore, and that is why such type of embryo sac is known as monosphoric embryo sac. Now in higher classes you will read this term Polygonal embryo sac. So what do you mean by Polygonal embryo sac? When the functional megaspore is at the chaliceal end and it divides further to form 7 cells and 8 nucleated embryo sac, then such type of embryo sac is known as Polygonal embryo sac. This is an important question for NEET as well. However, for grade 12 board examination, monosphoric embryo sac is very, very important. Thank you.